This section explains how to find the neurons that cause a behavior in a mammal. To learn which neurons cause a behavior, stop reading their activity and start writing it. The actuator is the experiment. Selectivity is bought by orthogonality. You install a foreign receptor whose trigger the surrounding tissue cannot answer. So one exogenous signal, light first, then a systemic drug, addresses only the cells you genetically chose. Wherever they systemic drug addresses only the cells you genetically chose, wherever they sit and however sparse. The chapter is the hunt for the cleanest such handle, a control channel present in your population and absent from everything else. Everything downstream is haggling over that one primitive, response speed, graded tunability, and avoiding an opsin synchronizing seizure. Causal dissection needs a lever. Nothing native can pull. This section explains the founding move of optogenetics, told through its first working instance. Selective control of neurons had always meant reaching the right place, an electrode planted in the right nucleus. The founding move of optogenetics inverts this. It makes the target a property of genotype rather than anatomy, and lets light be the trigger. The first instance is Zimmelmann's construct called charge. It is 3 Drosophila phototransduction proteins, arrestin 2, rhodopsin, and GQ-alpha, expressed together in mammalian neurons. Any cell carrying the transgene depolarizes under light, wherever it sits, whatever it wires to. The chapter is the field's origin story, told by the person who ran the seminal experiment. It opens the book by fixing the invariant that every later tool merely refines, genetic addressing, and optical triggering. It concedes at the same time the clumsiness that channel rhodopsin would soon erase. This section explains why a wiring diagram alone cannot tell you what a nervous system does, and how light-driven perturbation recovers the missing function. A connectome is an adjacency matrix without weights or signs. Knowing that neuron A contacts neuron B, even knowing how many synapses join them, never tells you whether the contact excites or inhibits, nor whether it matters for any behavior. Anatomy underdetermines dynamics. The move that recovers function is perturb and observe. Optogenetics writes controllable input into named neurons. Light-gated channels bypass the sensory world, while behavior and an optical calcium readout report the output. So the sign and weight of a connection fall out of the intervention, not out of the wiring. The worm? C. elegans makes this chi. Its body is transparent, so light enters freely. Promoter-specific expression makes each of about 100 central neurons individually addressable. And its fully mapped circuit of 300 O2 neurons is waiting to be functionalized. This section explains how the two-body concept turns a wiring diagram into a set of tested causal claims. A connectome is a map of what could talk to what. In the worm, it is 302 neurons, about 5,000 synapses, fixed since 1986, identical from one worm to the next. But an anatomical edge tells you only what could connect, never what the connection actually does. The wiring underdetermines the behavior it generates. Optogenetics supplies the missing operator. You express a light-gated channel in one identified neuron, illuminate the transparent worm, and read the movement. The light is the intervention. It promotes a graph edge to a tested causal claim, one node at a time. C. elegans is the substrate that makes this exact. It is transparent, so light reaches every cell. It is genetically addressable, one promoter per neuron, and it is small enough that every node is individually drivable. This section explains the single genetic move that the whole chapter is built on. A single light-absorbing protein dropped by genetics into one chosen cell makes that cell's membrane potential a function of illumination. This is the whole move, and everything in the chapter is a variation on it. Install channel rhodopsin and blue light depolarizes. Install a halar rhodopsin pump and yellow light depolarizes. Because the opsin is single component and gene targeted, you get a remote, cell-type specific handle on the causal chain from one neuron to whole animal behavior, the thing correlational recording could never give you. Kynorhabditis elegans is the chosen testbed. It is transparent, so light reaches every neuron, and it is wired to fixed 302 neuron synapse resolution, so the map from perturbation to behavior is legible. Distinct absorption spectra make each opsin an orthogonal control channel. This section explains optogenetics, the trick of turning a neuron's voltage into something you can drive from outside with light. Install a light-gated ion channel into any neuron you can name genetically, and light becomes that neuron's input wire. You express it wherever you silence with yellow, non-invasively, resolved to the millisecond. That is optogenetics. You borrow a microbe's photoreceptor, channel rhodopsin, taken from an alga's photoreceptor, channel rhodopsin, taken from an alga's phototaxis apparatus, you express it wherever you choose, and membrane potential turns into an addressable variable driven from outside. The generative move is transduction by installation. You couple an external carrier you control precisely, a wavelength, to an internal state you otherwise could not reach, voltage. C. Elegans is the ideal proving ground. It is transparent, it has 302 neurons wired to synapse resolution, so the light lands and the behavior reads straight out. Invert the same trick and you get sensors, with voltage or calcium reported back to you as fluorescence. This section explains how optogenetics is repurposed in Xenopus tail regeneration, where light stops being a trigger for spikes and becomes a knob on the bioelectric code. The chapter's move is to strip optogenetics of its neuroscience framing. No action potentials, no excitable cells. Instead, a light-driven proton pump, archaea rhodopsin, is used to set one scalar in ordinary cells, the resting membrane voltage, written Vmem. That single variable turns out to be instructive rather than epiphenomenal. Hyperpolarize the stump of a refractory stage Xenopus tail, and the whole appendage regrows, spinal cord, muscle, and vasculature together. The primitive to carry forward is that a complex morphogenetic program can be gated by one physiological parameter, so a single well-placed input reads out as an entire anatomical outcome. Optogenetics is here a voltage clamp, a push. This section explains how one experiment turns optogenetics from a tool for firing neurons into a tool for steering development. Optogenetics is usually a spike switch for neurons. Here, it becomes a voltage knob on cells that never fire. Adams, Tseng, and Levin express a light-driven proton pump, archaea rhodopsin, in Xenopus tadpole tails. They then illuminate an amputation made at the refractory stage, the window where the tail normally cannot regrow. Light drives protons out of the cell. It hyperpolarizes the resting membrane voltage, and the entire complex appendage regenerates. Here is the step that carries the whole result. Resting membrane voltage, Vm, is not a byproduct of physiology. It is an instructive morphogenetic set point, a command the tissue reads. So the chapter's role in an optogenetics book is to prove that the tool controls development, not only behavior. The controlled variable is bioelectricity, not the action potential. This part explains why light-driven sodium pumps were thought impossible and how one new structure dissolved that objection. A charged, translocated group seemed to forbid its own transport. The retinal shift base carries the positive charge that relays the proton, so shuttling a second cation of the same sign past it reads is electrostatically impossible. Hence the received wisdom that sodium pumps cannot exist. The chapter's move is to expose that as a hidden premise artifact. The impossibility was never a law. It was only the overgeneralization of one solved structure, bacteria rhodopsin, onto the whole rhodopsin family. Solving a single new structure, the Kr2 sodium pump collapses the no-go because the cation need not thread the shift base at all. In the book's arc, this hands Optogenetics its missing organ a route toward the outward potassium pump that neurons actually need for silencing. This section explains how a rhodopsin's function is encoded and why that encoding is a design key rather than a fixed fact of biology. A rhodopsin's cargo is written in 3 residues, same 7, helix scaffold, same retinal. What it pumps is set by which ionizable side chains line the active site, a proton for the DTD motif, chloride for NTQ, sodium for the NDQ motif of Kr2. That is the generative key. This chapter hands you. Read the motif, predict the ion, rewrite the motif, retarget the pump. So the optogenetics wish list, an outward potassium pump to silence neurons absent in nature, becomes an engineering search over that short key, not a hunt for a new protein. Here is the surprise it clears away. The field held that sodium pumps could not exist because every rhodopsin was assumed to be BR-shaped, a geometry where a positive shift base cannot shuttle a cation. Kr2 has a different geometry. It falsifies the assumption, not the physics. This section explains the single geometric constraint shared by all 6 methods for doing electrophysiology and optogenetics at once in a living brain. Every one of the 6 methods solves the same problem. The photon that drives an opsin and the contact that hears the spike must arrive at the same neurons. Optogenetics buys cell-type specificity at millisecond resolution. Extracellular recording is the most direct readout of what that manipulation did, but the readout is only valid where the light actually lands. So each method is one packaging of fiber plus electrode into a single implant. They differ only in how they trade illumination volume against recording proximity under a fixed tissue damage budget. Glued octrode, waveguide on shank, Insight micro LED, reversed fiber core electrode, optopatcher. The same move in every case. Co-registration of stimulus and sensor rotated through the geometry. The preparation forces. This section explains how a circuit reveals its causal structure. The key condition is that you can drive one node and read another at the same time, controllability paired with observability. That pairing is the whole of this chapter. Optogenetics is the right channel. Light leads to an opsin. The opsin drives membrane depolarization. The effect is specific to genetically marked cells and it is millisecond precise. Electrophysiology is the read, an electrode placed near enough to resolve spikes. Their simultaneous union is named SOG-P. That union is what converts a measured correlation into a demonstrated cause inside the living brain. Here is the generative move. Earlier tools forced a choice. Pharmacology was specific but slow. Electrical stimulation was fast but blind to cell type. Light collapses that trade-off, and the electrode is what lets you watch it land. This first section explains how optogenetics turns neural activity from something you watch into something you control, and what that reveals about axon growth. An electrode reads and writes at once, but it cannot say which fiber it drove. Light plus a genetically placed channel separates the two, making firing rate an addressable input rather than a confound. That is the whole move. Optogenetics converts activity from something you observe into something you observe into something you set, on the exact horizontal axons you set, on the exact horizontal axons you are already watching grow. Turn the knob and the axon answers. High frequency stimulation between 4 and 10 Hz halts a steadily elongating growth cone and collapses it. Low frequency does nothing. So, activity is not a generic fertilizer for wiring. It is a frequency-gated brake on growth. And you can predict that any activity versus morphology question yields to the same substitution. Convert the observable into an actuator. This section explains how the chapter collapses two operations that electrical stimulation had kept apart. Firing an axon and watching it grow become acts on the same object. A fine electrode perturbs an unknown population of fibers, so any link between activity and branching stays correlational. You never know which fiber you moved. Genetic addressing dissolves this. SHA-R2 lives only in the cortical neurons you transfected, so the light-driven axon and the imaged axon share one identity. Perturbation and observation now hold a common address. The causal claim that firing regulates horizontal connection growth bites on the very fiber under the lens rather than on a statistical shadow of it. This is developed in Chapter 8 by Malishevskaya and Yamamoto. This section explains the structural primitive behind an optogenetic tool that controls protein stability inside a living organism. Every optogenetic actuator is one move. A sensor domain undergoes a light-driven conformational change, and that change licenses the output of an effector domain. This chapter instantiates that move on the hardest-to-fake effector, destruction. The pieces compose like this. A dagron, the ODC-derived CODC1 tail that marks a protein for the proteasome, is caged inside the folded J-alpha helix of a LOVE2 photoreceptor. Blue light forms an FMN cysteine adduct, the helix unfolds, the dagron is exposed and activated, and the fused target is degraded. Protein amount then becomes a light-tunable equilibrium between constant synthesis and gated proteolysis, the half-life collapsing from about 120 minutes to about 20 minutes on illumination. The primitive is allosteric, uncaging. Light is the ligand, the helix is the lid, and degradation is the payload. This part explains the PSD module, a light-gated switch for controlling how much of a protein exists. Start from the accounting. A protein's steady-state level is a ratio, synthesis over degradation, so you can command its abundance without ever touching its gene, purely by opening a drain. The PSD module is that drain, and it is light-gated. A LOV2 photoreceptor keeps a destruction tag, the COD-C1 degron folded away in darkness. Blue light unfolds its J-alpha helix, exposes the degron, and the proteasome eats the fusion until a new, lower equilibrium settles. The generative primitive is conditional destabilization as a knob on the decay constant. It is reversible because biosynthesis refills the pool once the light stops. It is tunable because half-life sets the floor. And it is spatial because photons address territory. Fuse the cassette to the C-terminus of any nuclear or cytosolic target, and that target inherits light-controlled mortality. This section explains a class of light-switchable enzymes called photoactivatable nucleotide cyclases. The core object is a cyclic nucleotide cyclase made switchable by bolting a photosensor onto its front. The generative primitive is domain concatenation. A light-sensing module, either a BLUF domain or a LOVE domain, is fused to a catalytic module, either an adenylyl cyclase or a guanylate cyclase. So a photon's conformational nudge on the sensor propagates across the linker and gates catalysis. The enzyme is exactly what its two domains compose to. Light goes in, and a surge of cyclic AMP or cyclic GMP comes out with no wiring and no cofactor injection. Here is where the compression lives. Because the effector and the sensor are independently swappable, one photoreceptor scaffold becomes a whole actuator family, transcription, association, and signaling, all placed under light. The anchor is Euglenia's PAC alpha, and the bacterial version called B-PAX. Both respond to blue light and give second messenger control in the non-excitable cells that channel rhodopsin cannot reach. This part explains how a photoreceptor is built so that light can be handed to any downstream pathway. A photoreceptor is two separable domains bolted together. There is an N-terminal light sensor and a C-terminal effector, and the cell reads only their composition. That factorization is the whole trick. Hold the sensor fixed, swap the effector, and any downstream pathway falls under light control. The photoactivatable nucleotide cyclase, abbreviated PAC, is the clean instance. It is a light-sensing domain fused to a cyclase. A blue photon bends the sensor, the bend flips the enzyme, and cyclic AMP surges on cue. Optogenetics here is exploiting a modularity that biology already built. It treats sensor composed with effector as a swappable product, where you rewrite one factor and inherit the rest. This part explains how bioluminescence lets light-sensing molecules fire without an external lamp. Optogenetics pays a fixed tax. The photon arrives from outside, so every deep neuron needs a fiber physically threaded to it. The luminopsin, abbreviated LMO, cancels that tax by moving the lamp inside the molecule. The recipe is simple. You fuse a luciferase, the Gaussian enzyme known as G-LUC, to channel rhodopsin, then inject its substrate coelenterazine as an ordinary systemic drug. The enzyme's blue glow, peaking near 470 nanometers, sits right at channel rhodopsin's excitation peak, so it fires the opsin from within, everywhere the gene sits. One protein now carries two throttles. A fiber gives a sharp local pulse. A substrate gives a whole brain wash. The generative move is co-location. Set the source against the sensor, and the external delivery constraint that shaped the entire field quietly dissolves, folding chemogenetic reach and optogenetic specificity into a single handle. This section explains luminopsin and the structural move that makes it work. The real constraint in optogenetics is not the opsin itself. It is how light reaches the opsin. A fiber optic has to drill into the tissue, and it lights only a pinpoint. Luminopsin dissolves that constraint by moving the light source inside the molecule. Here is how. You fuse a luciferase to the opsin. Now, the trigger is luciferin, a diffusible substrate that crosses the blood-brain barrier after a peripheral injection and makes the enzyme glow right against its tethered opsin. The generative move is this. An actuator's input modality is a swappable part. Replace the photon emitter with a chemically fed one, and the same molecule inherits the affordances of chemogenetics, chronic, brain-wide, fiber-free control. And you keep physical light for the times you want it fast and focal. One actuator, 2 delivery regimes, chosen per experiment. This section explains why the path to the clinic for optogenetics is set by the delivery device and why epilepsy is the right first target. The therapeutic viability of a technology is decided by the binding constraint of its delivery device. Here, that constraint is power. An implant's longevity is drained by the light itself, not by its circuitry. From that single inequality, the whole chapter's ranking of indications falls out. The right first, in human target, is the one whose therapeutic logic tolerates the lowest duty cycle. On-demand seizure suppression, active less than 2% of the time, beats continuous deep brain stimulation, which runs roughly 50% of the time, which in turn beats indications whose schedule is unknown. Epilepsy wins by this criterion alone. The gap that keeps it hard is scale-up. Gross behavioral effects in rodents shrink to barely measurable saccade shifts in primates. The same opsin, a brain 2 orders of magnitude larger. This section explains when optogenetics crosses from the laboratory into the clinic. Optogenetics reaches the clinic exactly where a disease is, a circuit fault rather than a substrate. Rot, the chapter's move is this. The properties that made light, gated opsins a laboratory scalpel, millisecond on and off, cell type and projection targeting, bidirectional excite or silence chosen by wavelength, are the same properties a therapy wants against depression, obsessive-compulsive disorder, epilepsy, and essential tremor, where suffering lives in a mistimed or misrouted loop you can address. They buy nothing against neurodegeneration, storage disorders, or schizophrenia, whose pathology is diffuse and has no loop to seize. The generative reading is that a research instrument becomes a treatment only where its native affordance, here addressable specificity, matches the geometry of the failure. The new price is that the actuator is foreign protein in living tissue. This section explains why a tool built to control voltage was repurposed to control chemistry in astroglia. Optogenetics was built to drive neurons. Light opens a cation channel. The membrane depolarizes. The cell fires. Astroglia do not fire, so depolarization is inert in their signaling. The same actuator, channel rhodopsin 2, is redeployed by exploiting what was a side effect in neurons, the channel's minor calcium permeability. That trickle of calcium ions triggers the astrocyte's native chemical repertoire, its store-released calcium, cyclic AMP, lactate, and pH shifts, which is the real message. The surprise the chapter lands is that the initial influx barely matters. Downstream intracellular amplification does the work. A tool for controlling voltage is exapted into a tool for controlling chemistry because the target cell reads the side door, not the front one. This section explains why optogenetics, built for neurons, has to be re-engineered from the ground up for astrocytes. Optogenetics was engineered around one currency, membrane voltage, because neurons compute in spikes. Light opens a channel, the membrane depolarizes, and action potential fires. Astroglia break that frame. They carry a resting potential yet never spike, so depolarization signifies nothing. The chapter's thesis is a re-aiming. To control a cell optogenetically, you match the actuator to its own native signaling variable, the astrocytes rather than the neurons. For astrocytes, that variable is intracellular chemistry, calcium ions, cyclic, AMP, lactate, and pH. Even channel rhodopsin too, an ion channel, earns its keep through chemistry rather than voltage. A side door calcium trickle triggers internal store release, with autocrine ATP amplifying it. The generative move is this. Read a cell's currency first, then choose what the light perturbs. This part explains why oxytocin optogenetics needs two separate kinds of control, and how the chapter earns the second one. The usual promise of optogenetics is cell-type specificity. A short oxytocin promoter drives channel rhodopsin into only the oxytocin neurons, so blue light fires that one population and nothing else. That buys you control over which cells you excite. But peptidergic neurons broadcast. Their bifurcating axons spray oxytocin across many distant targets at once, so lighting the cell body gives tangled, unpredictable outcomes. Knowing which cell you fired does not tell you where the peptide lands. The chapter's real move inverts the target. Stimulate the far axon terminal rather than the cell body, and you fix both which cell releases and where it releases. Light placed on the spinal terminals of hypothalamic oxytocin axons drove pain, wind up down to about 40%, and that effect was abolished by an oxytocin receptor antagonist alone. Eliava and colleagues showed this in 2016. Here is the primitive. The promoter buys the source, and terminal illumination buys the site. These are two orthogonal specificities, and the peptide's diffuse anatomy forces you to control each of them separately. This section explains how a chapter turns the chemical identity of oxytocin neurons into optical control. Oxytocin neurons are chemically defined, not electrically distinct. No firing signature separates them from their hypothalamic neighbors, so classical electrophysiology cannot find them. The chapter's single move dissolves that problem. Splice a short fragment of the oxytocin gene's own promoter, roughly 0.1 to 1.9 kilobases, upstream of channel rhodopsin in a small RAAV. Inject the nucleus. Inject the nucleus. Only the cells that already transcribe oxytocin will transcribe the opsin. The promoter is the address. Specificity comes from what a neuron reads in its own genome, not from where the needle lands. Chemical identity becomes optical controllability at millisecond resolution. Every downstream method here, the slice, the anesthetized preparation, the freely moving animal, the long-range spinal projection, is just that one handle, turned. This part explains why optogenetics achieves cellular precision, and it names the single move that the whole toolkit reduces to. Start from the physics. Light is a broadcast. Every neuron sitting in the beam receives it. So the precision that optogenetics is famous for cannot come from the light itself. It comes from restricting which cells can hear the light. That restriction is written in the genome. A cell-type specific promoter, or the intersection of Cre recombinase with a lock-stop LOX opsin allele, decides which cells express a light-gated channel at all. The opsin then chooses only the sign of the effect. Channel rho, dopsin. 2 depolarizes. The chapter's whole toolkit is one move, addressing. Genetic targeting is the address decoder. The opsin is the actuator. Light is merely the enable pulse. So whenever you meet a new opsin line, ask first what promoter gates it. This section explains the real subject of the optogenetics body chapter, which is not the light switch but the problem of aiming it. Light hands you a universal actuator. Channel rhodopsin 2 opens on blue light, while halorhodopsin and archaeorhodopsin silence the cell. But a switch that fires any neuron at all is useless in an organ where every function rides a different cell type. The entire content of the chapter is the addressing problem. The difficulty is not the switch, but delivering it to exactly one population inside a heterogeneous brain. Every strategy the chapter surveys is one operator. There is the promoter, the Krelok's recombination system, the viral serotype, and the injection site. The craft is composing them, intersecting these predicates until only your target cells satisfy all of them at once. Read this way, the opsin is interchangeable. Specificity is the whole science, and each new recombinase, like DRE or FLP, merely adds another term to the intersection. This section explains how optogenetics changed what makes a neuron respond during circuit mapping. Older circuit mapping localized the stimulus. A focused uncaging beam freed glutamate at one spot, and every neuron there fired regardless of type. Optogenetics inverts the locus of specificity. You encode light sensitivity genetically in one cell type, you let the illumination go broad, and only the encoded cells transduce it. Connections that indiscriminate electrical stimulation blends together now become separable. The left and right, CA3 to CA1, projections intermingled at the CA1 synapse carry different plasticity, yet electrical stimulation recruits both and reports only their average. Select one input by its genetic address and the hidden structure resolves. This section explains how optogenetics changed the way neural circuits are mapped. Circuit mapping asks which neuron feeds which. The older method, glutamate uncaging, localized the stimulus. A focused beam rasters the tissue, and the input map is merely the record of where you had to aim to fire a presynaptic partner. Optogenetics inverts the addressing. Encode light sensitivity genetically, and the response is what gets localized instead. Illumination can be broad, and only the chosen cell type answers. As Zemelman and Miesenbuch put it, conventional photostimulation must localize the stimulus, whereas genetic schemes would localize the stimulus. Spatial specificity migrates from the beam into the genome. That single move is what the chapter is for. Every downstream capability follows from it. Cell type selectivity and mapping a projection's bare existence rather than a soma's position. This section explains how the chapter turns a correlation into a cause. The whole argument is a single conversion, correlation into cause. Schultz's electrophysiology showed that dopamine neurons in the ventral tegmental area fire in a way that tracks a reward prediction error. But firing alongside a signal can never prove that the firing drives it. Optogenetics closes that gap by adding two powers that electrodes lack. The first is cell type addressing, a Cre-restricted channel. Rhodopsin lights up only the dopaminergic cells or only the GABAergic cells and nothing else. The second is millisecond timing. With those two powers, you can test sufficiency and necessity. For sufficiency, phasic stimulation on its own conditions, a place, preference, or reinforces a nose poke. For necessity, inhibiting the neurons during reward. Omission blocks extinction learning. The reward prediction error turns from a description of what dopamine neurons do into the lever that writes behavior. This is the same move throughout the chapter. Every claim that these neurons correlate with something gets promoted to the claim that these neurons cause it. This section explains why the study of reward had to jump from watching neurons to controlling them. Electrophysiology could only watch. Schultz's putative dopamine neurons fired like a reward prediction error. But firing alongside a signal is not the same as driving it. The whole move of the chapter is the upgrade from correlation to control. Optogenetics turns a genetically defined population into a variable you can set. Here, the population is dopamine neurons given the light-sensitive channel GR2 through Cre recombinase expressed under the DAT promoter. Once they carry that channel, you can switch them on the millisecond timescale and read the behavioral consequence directly. So the reward prediction error stops being an observed signature and becomes a testable cause. The two questions become sharp. Does driving these cells produce reinforcement? Does silencing them abolish it? Part 4 opens on reward because it is the cleanest place to demonstrate the general primitive behavior explained by making its neural substrate an actuator you can set, a cause you drive rather than a signal you merely sense. This section explains why optogenetics changed how neuroscience dissects depression and addiction. Pharmacology turns a single scalar knob. A monoamine drug floods every synapse at once, so any behavioral change correlates with 100 cell types and is caused by none in particular. Optogenetics swaps the knob for an address. A Cre promoter selects one genetically defined population, an opsin makes it answer to light, and millisecond pulses toggle exactly those cells in a behaving mouse. Specificity is the entire move. Cell type plus timing upgrades the weak claim. This region correlates with reward into the strong one. Firing these VTA, dopamine neurons causes it. The chapter's payload for the book is a reframing. Depression and addiction are no longer chemical imbalance diseases. They become neural circuit disorders, dissectable one projection at a time, with the susceptible versus resilient split under identical stress as the sharpest proof. This part explains how optogenetics turns correlation into causation. It works by replaying, inside a living animal, the exact firing pattern a disease produces, then reading whether the behavior follows. The spine of the chapter, shown in figure 18.3, is a chain. Stress or a drug leads to a behavioral phenotype, which leads to a circuit adaptation, which leads to a channel mechanism, with light closing the loop. Drive the pathological VTA. Dopamine firing, and depression appears. Silence that firing, and depression appears. The whole method is that upgrade, from the claim this circuit is altered to the claim this firing is sufficient and necessary. The generative move transfers to every instance. A recording that merely correlates a variable with a state becomes a causal claim the moment you can set that variable to its observed value and watch the phenotype flip. This section explains why optogenetics is the pivot of the chapter. The single move is that it converts observation into intervention. Before optogenetics, behavioral neuroscience could record that dopamine activity correlates with reward. It could never show that dopamine causes the learning. Light-gated opsins change this. They let a chosen cell type fire at a chosen sub-second moment. The cell type is picked out by Krelok's spatial addressing, and the timing is picked out by temporal addressing. Together, these turn to spatial addressing, and the timing is picked out by temporal addressing. Together, these turn to specific neural axis Chefman. The anchor is Steinberg's blocking task. Injecting an artificial dopamine burst into the VTA at a moment where no prediction error should exist, manufactures learning about a redundant cue. This demonstrates sufficiency. The dopamine burst is enough to drive learning on its own, a claim correlation could never reach. That two-axis addressability is the primitive. Every reward circuit result is the same move aimed at a different pathway. The boundary is what sufficiency leaves open. Showing that a pattern can drive behavior is a weaker claim than showing the brain actually uses that pattern under natural dynamics. You have proven the pattern is capable, not that it is the mechanism the brain relies on. This section explains how optogenetics turns a known correlation about reward circuits into a demonstrated cause. Reward circuits were known only by correlation. Optogenetics converts that correlation into causation. The chapter's move is a single primitive. Match the resolution of the intervention to the resolution of the phenomenon. Reward-relevant activity is cell-type specific and sub-second. So a manipulation that lacks either axis cannot license a causal claim and both a lesion and a drug blur. One of them, the Krylok system, supplies the spatial axis, expressing light sensitivity only in the dopamine cells defined by TH, for example. Light supplies the temporal axis, defined by TH, for example. Light supplies the temporal axis, reads out sufficiency versus necessity. Here is the surprise that anchors it. The correlational picture says the circuit merely signals reward. The truth is stronger. Stimulating the dopamine neurons of the SNC can stand in as the reward an animal will work for. The circuit delivers reward directly. It is worth exactly one lever press. This section explains why optogenetics reframes epilepsy treatment as a control problem rather than a chemical or surgical one. Start with what a seizure actually is. A seizure is a control system failure. Excitatory drive outruns inhibition, and a focal patch of cortex runs away. The move of the chapter is to treat that runaway as an actuation problem, something you can drive back with the right switch. Optogenetics installs a light-gated switch in one genetically chosen population of cells. Because the switch sits in a single cell type, the same imbalance can be corrected two ways. You can hyperpolarize the excitatory principal cells, quieting the drivers directly, or you can drive the inhibitory interneurons that clamp those cells, tightening the brake. Against the incumbent therapies, optogenetics operates in a different regime altogether, not merely at a different strength. Drugs bias the whole brain. Open loop and reversibly, surgery deletes the focus, irreversibly. Optogenetics is reversibly. Optogenetics is reversible, precise to a cell type, and feedback-gated. A detector watches for runaway firing and feedback-gated. A detector watches for runaway firing and fires the switch only then. Therapy becomes closed. Loop control of a defined circuit. This section explains how one chapter recasts the treatment of epilepsy as a control problem. Epilepsy reduces to a single control failure. Excitatory drive outruns inhibition, and firing runs away. The conventional answers, drugs and surgical resection, respond bluntly. They are systemic, permanent, and blind to cell type. The chapter's move is to recast seizure suppression as closed-loop control. Here, optogenetics supplies the missing actuator, a photosensitive membrane protein. You choose its sign, either hyperpolarize the runaway excitatory cells or depolarize the inhibitory interneurons that gate them. You choose its target by promoter, which decides which cells the protein is expressed in. You detect aberrant activity and illuminate only the focus, only when needed. The generative primitive is this. A disease becomes tractable the moment you own both the sign and the address of your intervention. Every strategy the chapter surveys is just a way of filling those two coordinates. This section explains how lost motor function gets restored and why the chapter treats paralysis as a control problem rather than a wound to heal. Paralysis is a severed relay. The motor command chain runs from cortex to spinal motor neuron to muscle, and it fails wherever trauma cuts it, as in spinal cord injury, or wherever degeneration erases its nodes, as in ALS. in the mammalian central nervous system, nothing spontaneously regrows across the break. The chapter's move is to stop trying to heal the lesion and instead reconstitute controllability by two orthogonal routes. The first route replaces the dead neurons with stem cell-derived grafts. The second bypasses the dead link entirely with an optogenetic actuator, light-gated channels that let an external signal drive muscle directly. Restoration is reframed as installing a new controllable node rather than repairing the old wiring. Within this optogenetics volume, this is the chapter where the tool turns therapeutic. The two routes converge when the graft is itself made light drivable. This section explains why paralysis is a control problem and how the chapter solves it by replacement rather than repair. Paralysis is a broken control channel, not a broken muscle. A channel you cannot repair, you can instead replace. Traumatic spinal injury severs the command path from cortex to cord. ALS kills the motor neurons themselves. Neither regenerates, and no drug reverses either. So the chapter's move is to abandon repair and rebuild the actuator with a new addressing scheme. Graft stem cell-derived motor neurons carrying a light-sensitive opsin into the peripheral nerve, then drive muscle contraction optically. Light becomes the command line. The graft supplies neurons where the originals died. Degenerative primitive is substrate independence of control. A muscle needs a spike, not that spike's original source. So decouple the actuator from the failed controller and readdress it. This section explains how optogenetics restores sight in retinal degeneration. Blindness from retinal degeneration is a camera with a dead sensor. And a live cable, the photoreceptors die, but the downstream circuitry that carries and processes their signal survives largely intact. Optogenetics exploits exactly this asymmetry. Instead of repairing the dead sensor, it hands photosensitivity to cells that never had it, transfecting surviving neurons with a light-driven extus. The constraint that governs the outcome is where, in the surviving chain, you install the new receptor. The closer to the dead photoreceptors you inject sensitivity, the more of the retina's native parallel image processing still runs on the signal before it reaches the brain. That native processing is more than a dozen feature-extracting microcircuits, and the more of them the signal passes through, the higher the restored extracting microcircuits, and the more of them the signal passes through, the higher the restored acuity. The approach is mutation. Agnostic by construction. One channel treats thousands of distinct diseases causing mutations because it bypasses the broken gene entirely instead of correcting it. This part explains why retinal degeneration takes away sight and what the repair strategy does about it. Blindness from retinal degeneration is a loss of the input transducer, not the wire. Photoreceptors, the cells that convert photons to voltage, die. The downstream processing network, the bipolar, amacrine, and ganglion cells, survives largely intact. The chapter's move is to stop trying to regrow the dead detector and instead reinstall photosensitivity somewhere else. You virally deliver a light-gated protein into a surviving layer, promoting those cells into new photon detectors. Vision restoration becomes a relocation of the transduction step further down the circuit, trading anatomical fidelity for a functional's the transduction step further down the circuit. There is a trade-off in where you aim. The deeper the layer you target, the more native processing you inherit for free. But the deeper you go, the more you must recreate, because you have bypassed the retina's own front-end computation. This section explains how optogenetics restores vision after photoreceptor disease and why the shift from traditional to designer tools is really a shift in sensitivity. The disease kills the transducer but spares the circuit, The photoreceptors die, yet the inner retina survives and still wires to the brain, so recovery works by substitution rather than reconstruction. You install a fresh light sensor one layer upstream in the ON bipolar cells, and you let the retina's intact processing do the retina's intact processing, the retina's intact processing do the rest. The traditional to designer axis is really a gain axis. Channel rhodopsin gates ions directly, a handful per photon, which demands snowfield bright, tissue-toxic light. Mammalian opsins are G-protein coupled receptors, so one photon fires many G-proteins, each catalyzing downstream. That buys 2 to 3 log units of sensitivity, each catalyzing downstream. The generative move is to see sensitivity as a catalysis problem rather than a bigger channel problem. Borrow the amplifier the cell already owns. This part explains how one chapter restores vision after photoreceptor death and why it refeeds the retina rather than replacing it. Blindness from photoreceptor death leaves the rest of the retina alive, its signaling machinery intact, merely unfed. The generative move is this: do not replace the dead pathway, refeed it at the top. A foreign light-gated channel, called HR2, forced into a surviving neuron, must do everything itself, so it needs snowfield in sunlight intensities. It is insensitive, toxic, non-physiological. The chapter's answer, called Opto-mGluR6, is a chimera. Melanopsin's light-sensing body is spliced onto the intracellular tail of mGluR6, so a captured photon hijacks the cell's own idle G-protein cascade, the native amplifier that turns one photon into hundreds of gated channels. Sensitivity, correct polarity, and on-off structure all come free because the cell's pre-existing machinery does the work, except the surviving substrate. Supply only the missing input. This section explains a strategy for restoring vision after photoreceptor death. When the photoreceptors die, sight is lost, yet the optic wiring downstream of them survives intact, so the repair target shifts to the neurons that remain. Express a microbial opsin in any surviving retinal neuron, and that neuron becomes an artificial photoreceptor, with light directly gating an ionic current across the membrane. Acting downstream of the lesion is what makes the fixed substrate agnostic. One construct answers all of the roughly 3,000 RP mutations at any stage of the disease. Gauvain calls this one size fits all. The structure of the approach shows in its cost. A single light-gated protein stands in for the healthy retina's two-stage amplification cascade, so gain and dynamic range nearly vanish. That forces the use of bright light and biomimetic goggles. The principle is to confer light transduction wherever the circuitry still lives. This section explains how optogenetics restores vision by working around a dead sensor rather than fixing it. Retinitis pigmentosa and macular degeneration kill the photoreceptors but leave the rest of the retinal circuit standing. The wiring survives, only its input transducer dies. Optogenetics exploits exactly that. You express a light-gated microbial opsin in a surviving downstream neuron, and it becomes an artificial photoreceptor, a single protein standing in for the entire phototransduction cascade. The dead sensor is bypassed, not repaired. The whole design then collapses to two choices: which opsin and which cell. The eye's transparency makes the target uniquely reachable by light. Here is the structural trade-off that seeds the chapter. The deeper into the surviving circuit you install the new transducer, the more of the retina's native amplification and dynamic range you inherit rather than rebuild. This section explains how holographic optical interfacing restores sight by writing the retina's output layer directly. The method begins by making the surviving ganglion cells light sensitive. You photosensitize them with channel rhodopsin 2, then you paint spike patterns onto them with light. The dead photoreceptor layer, the rods and cones, is skipped entirely. The generative move is holographic. A phase spatial light modulator diffracts the whole beam into the spots you want, rather than masking out the spots you do not. This is why selectivity costs no power. A mirror array throws away its off pixels, discarding that light. A hologram redistributes them instead, sending every photon somewhere useful. The payoff is efficiency. 1 milliwatt then lands on thousands of points at once. That yields single cell, millisecond, massively parallel control across a millimeter of retina. A camera feeds the hologram in real time. The primitive to carry forward is this: do not block light. Compute where it should go. This part explains how to restore sight to a blind retina and why one optical trick makes the whole approach work. Start by making the surviving ganglion cells directly light sensitive. Once you do that, the problem collapses to something simple: painting patterns of light onto individual cell bodies. The entire chapter is then one move: shape the light by its phase rather than by blocking part of it. A spatial light modulator bends the laser's wavefront so that it diffracts into exactly the target points. This is a computer-generated hologram rather than a mask that throws the off light away. The power stays the same, redistributed rather than subtracted. That lets you excite thousands of cells at single-cell resolution with about 1 milliwatt in 3 dimensions, with about 1 milliwatt in 3 dimensions, without any scanning. That efficiency is the surprise, and it is why holography wins over the two rivals. The electrode array blurs because its current spreads. The digital mirror device wastes most of the beam. Holography does neither. This section explains how the chapter restores vision in retinitis pigmentosa by turning a surviving neuron into its own light sensor. Blindness from retinitis pigmentosa kills the photoreceptors but spares the wiring behind them. The chapter's move is this: do not rebuild the dead sensor's machinery, rhodopsin, the G protein, phosphodiesterase, the whole C, G, MP cascade. Instead, install a single protein that is at once photodetector and actuator, placed into a cell, single protein that is at once photodetector and actuator, placed into a cell, still connected to the brain. Channel rhodopsin absorbs a photon and opens as a cation channel in one step. A multi-stage transduction chain collapses to one gene. Delivered by AAV into surviving retinal ganglion cells, that cell becomes its own light sensor and feeds the intact optic nerve directly. The primitive is this: when a sensory front end fails, but its downstream circuit persists, photosensitize the survivor rather than resurrect the cascade. This part explains how sight can be restored after photoreceptors die. When photoreceptors die, the retina's downstream wiring survives, so sight returns by moving sensing past the fault instead of resurrecting the dead sensor. Native phototransduction is a fragile relay of many proteins. Rhodopsin leads to G protein, leads to PDE, leads to channel. Re-expressing that whole cascade in surviving cells means orchestrating surviving cells, means orchestrating surviving cells, means orchestrating several genes at once. Which stalls? Channel rhodopsin collapses the chain into a single molecule, light-gated in cation, selective in one protein, sensor and channel fused. Transduce that lone gene, CHR2, or the lab's MVCHR1, into surviving ganglion cells, and each relay neuron becomes its own photoreceptor. The primitive is this. When a multi-stage transducer breaks, don't repair the stages. Graft. One self-contained component downstream of the break. This part explains why optogenetics changed memory research, and it walks through a single circuit that proves the point. Memory research already had its molecules and its correlations. What it lacked was a way to act on the timescale neurons actually run, milliseconds. Optogenetics supplies exactly that, and the whole argument of the chapter turns on that match. Intervene at the phenomenon's own rate, and correlation becomes causation. The demonstration is the AC to CA projection, a direct top-down pathway from anterior cingulate to hippocampus. A brief pulse of optical stimulation on that pathway is itself sufficient to make a mouse retrieve a fear memory. Turning the circuit on turns the memory on. The revealing contrast is in the silencing. Slow pharmacological silencing spares. Recall, because the brain recruits compensatory circuits during the lag. Millisecond optogenetic silencing abolishes recall because nothing has time to compensate. A circuit is defined by what its activation does. The loop closes only when your knob turns as fast as the thing you are turning. This part explains what it means to define a memory circuit by its own timescale. A memory circuit is defined by what silencing it does at the circuit's own timescale, and the chapter's move is to make perturbation and observation simultaneous, closing the loop between them. The anchor is Gaussian's inversion. Silence, CA1 slowly, over 30 minutes, and the anterior cingulate compensates. Recall survives, the region looks dispensable. Silence it in milliseconds, and recall collapses. Same neurons, opposite verdict. The rate of intervention, not the target, decided it. So causation demands a clock matched to the system's own dynamics before homeostatic rescue masks necessity. Optogenetics supplies that clock, and this chapter aims it at the neglected top-down direction, prefrontal cortex driving hippocampus, inverting the canonical bottom-up entorhinal frame. This section explains why the resolution of your probe determines which causal roles you can even see. Lesion and pharmacology are blunt instruments. They disable a whole region at once, so opposing cell types within it cancel each other out, and slow compensatory rewiring masks a structure's true function. You conclude that a structure is not necessary when the honest verdict was that it was never cleanly tested. Optogenetics swaps that for cell type-specific millisecond control. Under the finer instrument, a structure previously filed as mere output, the thalamus, the end root of arousal, resolves into a controller. Thalamocortical spindle bursting from the reticular nucleus gates the transitions between NREM and REM sleep and sets the arousal threshold, actively shaping sleep and sets the arousal threshold, actively shaping sleep architecture rather than relaying someone else's command. Sharpen the manipulation, and a relay becomes a governor. This section explains what optogenetics adds to the study of sleep, and how a sharper instrument can flip the direction of a causal claim. Optogenetics buys one thing that lesions and drugs never could: sufficiency, tested at a single cell type on a millisecond clock. Lesions read only necessity, and their slow compensation masked what a region actually does. So the sleep-wake map fossilized around the centers that were easy to ablate, with cortex and thalamus filed as the end root, the downstream output of hypoth with cortex and thalamus filed as the end root. This chapter's move is that finer causal resolution does not merely sharpen a map, it can reverse an arrow. Selective, timed control of the thalamocortical loop reclassifies it from a relay into a modulator of sleep architecture itself. The primitive is this: a beta instrument promotes a node from effect site to cause. This section explains why a cochlear implant hits a resolution ceiling and the one substitution that lifts it. A cochlear implant fails not for want of electrodes, but because its medium betrays them. Current injected into the conductive perilymph spreads and overlaps, so 24 electrodes collapse to 4 to 8 independent channels, and the roughly 30,000 tonotopically tuned spiral ganglion neurons stay coarsely addressed. The chapter's whole thesis is one substitution. Swap the stimulus carrier for one. The medium cannot smear. Light, unlike current, focuses to micrometers, so channel count is recovered by physics rather than fought by engineering. The generative move is this. Resolution is set by your stimulus's point, spread in its medium, not by emitter granularity. That is why light in place of electricity is the lever, and why the deeper current-drowned ABI stands to gain most. This section explains why restoring hearing through an implant is fundamentally a bandwidth problem. The bottleneck is not how many electrodes you place. It is how many independent channels they can carry. Injected current spreads through conductive tissue, so neighboring contacts excite overlapping neuron populations, and their frequency channels bleed together. The chapter's thesis is a carrier swap. Replace electricity with light. Light can be focused where current cannot, so the same anatomical target yields more separable channels, sharper tonotopy, and less off-target activation. That off-target activation is the acute failing of brainstem implants placed near non-auditory nuclei. Everything downstream, better speech in noise, localization, music, follows from that single resolution gain. Optogenetics is here, the instrument that buys spatial confinement. This section explains why the cochlea was the first nervous system prosthesis to succeed, and what optogenetics changes about it. Frequency lives as position in the cochlea, a line where pitch maps to place, so restoring hearing reduces to addressing points on that line independently, which is why cochlear implants succeeded first among neural prostheses. Their limit is the stimulus. Electrical current spreads through tissue, so one electrode excites a whole neighborhood of the map and the channels blur. Light spreads far less. Focus it, and each tonotopic point answers alone. Optogenetics is that substitution. Swap a leaky field for a confinable one and recover the channel count the anatomy already offers. The retina, lacking this clean one-dimensional map, needs hundreds of independent pixels. The cochlea's linear tonotopy is what made it the easy, already won case. This section explains how to address a single neuron class when no clean genetic key exists. No single gene names. One neuron class, so a cell type cannot be addressed with one key. In the primate, the genome is uneditable. The only vehicle is a virus, whose tiny genome cannot even carry a natural promoter's full address. Zemelman's move is to stop copying addresses and start computing them. He mines regulatory motifs conserved across distant species, synthesizes a minimal promoter from them, and then, because each synthetic key stays leaky, stacks two of them virally and intersects their overlap. Specificity becomes a Boolean AND of imprecise markers, a conjunction instead of one exclusive label. The primitive is this. When no single coordinate is unique, the conjunction of loose coordinates pins down what none of them can, alone. This section explains the single structural move behind Fallon's chapter on amino acids. It overturns one frame, the amino acid as an inert building block. The generative move is to read a single scarce resource at every level it operates on, so that its shortage propagates across all of those levels at once. The essential amino acid pool is three things simultaneously. It is a substrate, the raw material for proteins, and the precursor of dopamine and serotonin. It is a signal, feeding a deficiency sensing response system. And it is a switch, exerting epigenetic control over 500 or more genes, where starvation up regulates or down. Regulates or downregulates transcription. One example is CHOP-gating neural pruning. So one depletion cascades upward from metabolism to gene expression to phenotype. That single upstream scarcity binds autism, Parkinson's, schizophrenia, and cocaine addiction, the last running from phenylalanine through delta phos-B as downstream symptoms of the same shortage. This part explains how a single amino acid works as two things at once, and why its scarcity is itself a signal. One molecule serves two roles. An amino acid is the substrate a protein is built from, and at the same time it is the signal gating whether genes transcribe at all. Scarcity itself carries information. Consider two cases. Methionine starvation induces CHOP, which prunes neurons by apoptosis, and autistic brains, being overneuroned, read as pruning that never fired. Depleted phenylalanine, acting through delta phos-B, elongates dendritic spines after cocaine. So Fallon dissolves the building blocks cliché. The amino acid pool is a regulatory input, and its depletion composes disease pathology. The same lever this optogenetics book pulls with light, governing a system through one controllable input, is pulled here metabolically, with the input's mere absence as the actuator. This section explains why a single technical gain moving from electrical to optical brain stimulation generates the entire ethical picture for both the promise and the hazards. Electrical deep brain stimulation is a blunt instrument. One current floods a whole region, exciting axons while inhibiting cell bodies, activating glutamatergic and GABAergic neurons alike, so the opposite effects partly cancel each other out. Optogenetics buys specificity instead. A chosen wavelength of light. The chapter's central move is that this single gain generates the entire ethical ledger on both sides at once. The specificity is the whole clinical promise, and it is also the source of the new liabilities, gene therapy delivery, opsin toxicity and immune reaction, stored brain activity data, and the question of mind privacy. Mella and Walter's own verdict is honestly empty. The technique is untested in humans, so neither its risk nor its benefit can yet be specified. The real thesis of the chapter is therefore about method, bioethics done ahead of the technology, grounded in fact rather than in science fiction. This section explains why the chapter judges a therapy before it exists, and how the very thing that makes optogenetics precise also makes it dangerous. Ethics run ahead of the artifact. The chapter judges a therapy that has never touched a human on the principle that the responsible moment to evaluate a technology is before it exists. The move that makes this tractable is a contrast between two ways of stimulating the brain. Electrical deep brain stimulation fails by non-specificity. It floods a whole region, simultaneously activating glutamatergic cells and inhibiting GABAergic cells, whose effects then cancel. Optogenetics fixes precisely this. It selects the cell type and the sign of the effect by wavelength. Yet that same selectivity is the hazard. The knob that grants therapeutic precision also grants control, precision, unwanted personality change, self-alienation, and stored brain activity used as mental surveillance. So benefit, risk, and autonomy are weighed against empirical facts, keeping the judgment anchored in what is known rather than in science fiction. The cure and the danger ride one axis.